All right, so this video is going to focus on signaling pathways, but it's not going to focus on this huge system level response to stimuli. Start looking at like negative feedback and everything. What it's going to focus on a little bit more is more of this molecular level or cellular level response to stimuli. So as we get into this, when we get to the target cell, we focus on what's actually happening at that molecular level. So we specifically typically focus on reception, transduction, and response. So at the reception, we have our hormone and it's binding to the hormone receptor. Okay. Sometimes it can be environmental stimulus that's causing that. We have our reception. Then we have the signal transduction, and that's a lot of what this whole video is going to focus on. Many examples of signal transduction pathways, phosphorylation cascades, and stuff like that through secondary messengers, activating enzymes and all of that. And then we eventually get a cellular response. So sometimes that might be turning on DNA to make certain proteins which change things. Sometimes hormones turn on enzymes, and enzymes end up doing a series of chemical reactions within the transduction pathway, but then ultimately doing a final chemical reaction. Sometimes, like in the case of ligands and neurotransmitters, we can also have um, hormones that are opening ligand gated channels and letting things in. Sometimes this cascade of enzymes can also embed channels into the membrane. But again, what you're going to see here is a lot of complexity when it comes to these signal transduction pathways. And why in the heck why is it so stinking complicated? For one reason, it's amplification, which means it amplifies the effect of a signaling molecule. So one molecule can bind, but then you can get a big increase in that effect. Even though it's just a single molecule, you can have many things happening within the cell. In addition, complexity allows for control. Now, you might also think that complexity allows for more things to go wrong, which is kind of true as well, but it also gives the cell more control over the accuracy of the pathway. They have to all be functioning properly, and there's a smaller chance um, that the transduction might occur in error. Um, the final part is multiplicity, and that's different from amplification because multiplicity means that a single molecule can activate multiple other proteins, which all generate a different response. So multiple processes can be turned on by a um, sing, single reception. So this model here demonstrates multiplicity. So even though we have a single first messenger and a single receptor, we can follow this pathway. That receptor can activate other receptors, which activate other G proteins, which activate other enzymes, which ultimately trigger a cyclic AMP to do, like, for example, glycogen synthesis. But then you can also, from this single reception, trigger a different pathway, and this represents the multiplicity. So we can get different enzymes being activated, which causes different messengers and then different responses, which may be like smooth muscle relaxation. Ultimately, this one messenger also amplifies. So we have multiplicity because we have different responses from a single receptor, but you'll also have many more final messengers that have been activated as a through amplification. And this amplification is also shown here. So you can see, like, maybe we start with one molecule that's doing the recepting, binding of one thing to a receptor. But then we have this transduction, and transduction is many secondary messengers. So as we read through this, we can see that we've got this inactive G protein, which becomes active. We have an inactive adenylyl cyclase, which becomes active. We have ATP, which is transformed into cyclic AMP. You have inactive pKa, which becomes active pKa. You have inactive phosphorylase kinase, which becomes active phosphorylase kinase. You have inactive glycogen phosphorylase, which becomes active glycogen phosphorylase. And as you know this, you see as you go down here, the number of molecules activators increase, 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 and that's amplifying the signal. And eventually, the response causes a huge response, and as opposed to one molecule can cause a tremendous amount of molecules to respond. Other things I want you guys to pay attention to as you watch this video is the word kinase and phosphorylation. Now, we talk about within energy ATP, and as we release energy, we um, we break that final bond in the ATP molecule, and that phosphate causes a conformational change, which causes something to move, something to happen. So when it talks about inactive becoming active in that phosphorylation cascade, it's dealing with a lot of kinases. So what the heck is a kinase? It ends in A, so it's an enzyme. It modifies other proteins by chemically adding phosphate groups to them, which is phosphorylation. So ATP is going to be involved, or GTP. But phosphorylation usually results in a functional change of the target protein by changing its activity, location, or association with other proteins. So in the human genome, there's about 560 different protein kinase genes that constitute 2% of all the human genes. So these are a pretty significant type of protein. And up to 30% of all human proteins may be modified by kinase activity. So a third of all proteins can get regulated by attaching phosphates to them. And this is what I want you to kind of picture as you see all the phosphorylation happening. It's inactive right now, but let's say this is the phosphate group. When the phosphate attaches, it creates a conformational change here, which now allows things to actually fit into the active site and be go through a chemical reaction here. So it's pretty significant, and it's especially involved in signal transduction. Now, one other thing, phosphorylation cascade. You guys, sometimes this is referred to as a cascade, and you see how this kind of that cascading like a waterfall, that's where it gets that name because it's one is triggering the next, it's triggering the next, triggering the next until we get to the response. So the first example we're going to look at here involves the pancreas. So um, where the heck is your pancreas? I mean, you hear about it with um, insulin and those kind of negative feedback mechanisms. If we follow our digestive tract down here, where I'm at right here, this is the esophagus and it's going into the stomach. If you would cut something open, the liver would kind of lay on top, and its liver is huge. And then the stomach ends up emptying into the duodenum and the small intestine, which eventually leads into the large intestine or the colon. Now, the pancreas is kind of hidden right in here. And the pancreas is important because it releases hormones, but also certain digestive enzymes get secreted into the digestive tract as well. So the pancreas is really significant, especially when we start thinking about insulin and glucagon, and those are two of the examples we're going to look at here. So again, you guys don't have to worry about every single word here, but I want you to see multiplicity, and not multiplicity, amplification. And I want you to see kinases, and I want you to see phosphorylation cascade. We're really only showing one response here, so we're not showing all of the multiplicity, but the reality is it's complicated enough as it is, but I could show because like PKB here, that does, it triggers other things as well. So besides just the release of these um, GLUT4 channels. So insulin, it's a hormone. It's a hormone that lowers your blood sugar. So what these all represent are sugar in your blood. Glucose is in your blood, and it reduces blood sugar because it triggers a cell here to allow the glucose to come in. So the insulin's going to bind. We're going to cause this chain reaction of transduction pathways. Look at all this ATP here. So all of this ATP is the source of the phosphates for the phosphorylation cascade. Ultimately, we're going to get these channels into the membrane so that the sugar can come in. So another thing I want you to focus on here is control. That signal transduction pathway, in addition to amplifying a response, all of those complicated steps provide for control. All components of the pathway must be functioning properly. There's a smaller chance that the transduction might occur in error. So we start with insulin. It binds to the receptor, which causes it to dimerize. And now from there, we have all of this ATP phosphorylating the inside part of the receptor, which causes the phosphorylation of the next kinase. And then that can move on to this PIP2, which causes it to be PIP3, which causes the next kinase to bind, which causes the next phosphorylation. And this is going to move over here and phosphorylate this cap, which occurs on the vesicle. So these proteins are made and put into the membrane to this vesicle. So these GLUT4 proteins, these channels, are made through the endoplasmic reticulum when we get into that all that protein synthesis business. But again, this kinase helps phosphorylate the cap, which causes it to be removed. When the cap is removed, this vesicle can attach to the um, kinesin motor proteins. And again, this is just part of the cytoskeleton protein. And now we can use ATP energy to move the kinesin motors. And ultimately, we embed those GLUT4 proteins channels into the membrane, which allows sugar to diffuse into the cell. And therefore, through all this transduction pathways, we end up having insulin causing a response, which is lowering blood sugar. Now, that's a lot of steps, right? So if you are explaining diabetes to an elderly patient who hasn't been in biology class for 50 years, you might not take them through that series of reactions. But when you look at this scientific model, this may not be 100% scientifically accurate, where it just shows like the insulin binding to the receptor, and then that just opens the channel, and then the glucose comes in, right? Insulin is the key that unlocks the glucose channel. So once it lands, then you can let stuff in. Now, that's a lot easier to understand to someone who hasn't been immersed in signal transduction pathways. It may be less scientifically accurate, but all scientific models are a balance between accuracy and understanding. So, again, looking at this model, is it really scientifically accurate? Obviously not. You don't actually take a key and open a door, and then the sugar comes in through this huge gaping hole. If you opened a hole like that in a cell, the cell would die. So, but at the same time, can this scientific model be helpful? Yeah. Okay, so how about this one? The difference between type 1 and type 2 diabetes. In the case of type 1 diabetes, there's no key or no insulin to unlock the door. If it's type 2 diabetes, you have insulin, but it's unable to unlock the door properly. Or the key is there, but the lock doesn't work. So you see how there's no hole in this one? So type 2 diabetes, you're producing insulin, but your doorways don't open. So is that really 100% accurate? Not quite. But it does. Scientific models are a way to make the world easier to understand. Again, same idea here. You've got normal cell, insulin binds to the receptors. Now, are the receptors and the channels connected on the same molecule? No, it's not like a ligand-gated ion channel, like in neurotransmitters and nervous system communication. But this still makes it a little bit easier to understand. So if it's insulin resistant, your insulin receptors aren't receiving the insulin. The insulin is there, but your blood sugar stays high because you're not able to get enough channels or receptors to work properly. So again, may be less scientifically accurate, however, it can still be helpful. Now, this pathway with every single tiny little molecule labeled is probably a lot more accurate than mine. However, would this be helpful to explain to a diabetes patient who hasn't had a biology class for 50 years? Probably not. But as we keep on looking here, if you're insulin resistant, where is the problem with the pathway? It can be an incredibly complicated scientific task to understand like what is actually wrong. So I want to go through a couple more examples. I'll try to keep this thing moving. But again, as we go through this, continue to focus on um, the signal transduction pathways are advantages because they can amplify the effect of a signaling molecule. All of those intermediate steps provide control, and you can also take one single molecule and have it have multiple effects. Again, look at the cascade. G protein, adenylate cyclase, ATP turning into cyclic AMP, protein kinase A. And one thing triggers it once it becomes activated, it can phosphorylate, it allows for the phosphorylation of the next kinase, and we trigger all these inactive enzymes becoming active. So this one is going to focus on antidiuretic hormone within your kidneys. So I know we don't do a lot of anatomy in here. I think it's important to understand some of the major biological processes. So what the heck do your kidneys do? They filter your blood, okay? They do end up producing urine, but as they do that, they're taking nitrogenous waste out of your blood. All of those chopped up amino acids, all that amine groups and urea gets taken out of your blood through your kidneys. So if someone's kidneys aren't working, they need dialysis, which what does the dialysis do? It filters your blood. And blood is taken out and move back in after it gets filtered. So as we look at this, we break this down, we get zoomed in, and we have blood coming in, and it's from the heart to the kidney. And then these things go into these glomeruli, which does the filtering. And then only the biggest things stay in the blood vessel here. Everything that's small, including most of the water, um, gets put into these uh, Bowman's capsules here. And in through this descending loops and the um, convoluted tubules, and ultimately this would lead to your bladder to your ureter and ultimately to your bladder. But as it goes through this, it's not selectively filtering here. Just everything that's small gets punched out. Well, some of that small stuff we want back, so we do filter back into the bloodstream here, and it the good stuff that we want back comes in and gets taken back towards the heart here, from to these veins back to the heart, which includes a lot of water. We don't want to pee out all of our water, so we have antidiuretic hormone, which triggers more diffusion of water from these um, loops of Henle back into your bloodstream. So again, we keep on zooming in. We got kidneys, we got blood coming in, getting filtered here. Um, all of the urea is going through your ureter, and ultimately collecting as urine in your bladder that you pee out. But in that process, the glomerulus here is where we're having all of that little stuff get pushed into these proximal convoluted tubules and the loop of Henle and all of that kind of stuff. So in that loop is when we do a lot of the reabsorption. But as we keep on looking here, again, how does that filtration work? These are cells wrapped around blood vessels, and they're tiny little pores. So it's really mechanical filtration, which is pretty amazing. And I mean, we could spend a whole time talking about kidneys, but let's get into the pathways. Again, focus on kinases and phosphorylation. So where do we see ACEs? These are enzymes, right? We have ATP, which will become cyclic AMP. We have an aquaporin channel. We have all of these aquaporin channels here being kind of held back in this cage. And we have water that's already been filtered out. Now, again, we don't want to pee out all of the water, so we want to bring that water back into our blood. So at this point, it's already been filtered, and we have these individual cells to try and bring the water that's about to lead to our bladder, bring it back into our blood. So along comes ADH. It's a hormone. It binds to a receptor. GDP is replaced by GTP, which causes this G protein to slide along the cell membrane, which activates the adenylyl cyclase, which turns ATP into cyclic AMP, which then activates another kinase, which then removes this cytoskeleton net, which allows the aquaporin channels to go into the membrane, which then the water will diffuse through the cell and back into the blood. And that's how ADH works. So, another idea to think about here is, how do drugs work? And sometimes we take drugs, like prescription drugs, to help um, if this process isn't working properly. But sometimes drugs act as inhibitors. And we're going to focus on alcohol as a drug and how it works as a diuretic, causes you to pee more or have more liquidy pee. So we can have competitive inhibitors that bind directly into the active site. And again, these are enzymes here. It's going to be competitively inhibitive of the receptor. But it's the same idea. We have proteins, everything communicates through shape. If it blocks a shape or changes the shape in some way, it can cause things to not work. If something is inhibited, it's not functional. So you can see my little alcohol molecule here and ADH. So if alcohol inhibits the antidiuretic hormone, it's considered a diuretic. What's going to happen next? It changed the shape here. Is antidiuretic going to be able to land? The answer is no. So if it can't land, all of this transduction pathway does not happen. The water can't get pulled back through these aquaporin channels and ultimately back in the blood. So what happens? Your pee is really liquidy, okay? Because the water was not reabsorbed. It got filtered out of your blood, but then it didn't get brought back into your blood because ADH was not able to send the communication in. We didn't have a reception. If we don't have reception, we don't have transduction. If we don't have transduction, we don't have response. So again, that gets back into this whole thing. When we look at cellular molecular level, we're focusing on reception, transduction, and response. Okay, one last one, glucagon. It's doing the reverse of reverse response of reverse effect of insulin. That doesn't necessarily mean it works the exact same way. And glucagon is going to, this is a quote taken from something else, when there's no food intake, glucose decreases, your blood sugar goes down. Glucagon is then secreted from the pancreas to signal the liver to produce glucose. Well, how does that work? The liver produces glucose? What in the heck does the liver produce glucose? I thought we ate carbs and plants produce carbs. What does the liver have to do with carbs? A lot of times people think about liver, they associate it with alcohol and cleaning up toxins, which is true. But the liver is amazing. I mean, it makes all of the proteins within your blood, all the plasma proteins, but then it has a lot to do with communication, and it also stores excess carbohydrates in the form of glycogen. So, again, point of reference, we were talking about the pancreas before. The liver sits here. Um, It also makes bile, which is stored in the gallbladder, which helps in the digestion of fats and isolating the fat, so lipases can break down those lipids. But I mean, we could spend another half an hour just talking about the liver, but let's move on. So, again, all of this from your digestive system, before, as you digest all of your food here and all these nutrients are coming back, all those nutrients run through your liver prior to getting pumped up to your blood. So if you have a lot of excess carbohydrate that diffuses into our bloodstream, it'll top off the liver, and that glucose is going to go into the liver and get built up into glycogen to be stored. Now, if our blood sugar gets low because we haven't eaten in a while, we have all this stored up glycogen to put the glucose back into our bloodstream. And again, all that blood is diffusing into all of these individual cells within your liver. So, how does it work? We got glucagon, which is a hormone. And we've got this big transduction pathway. Again, this is a G protein receptor, so we're going to get um, adenylate cyclase turned on. And again, you see kinases, you see ATP, which means we're going to have phosphorylating happening, where we activate certain enzymes by adding phosphate groups to them. Ultimately, we have this big stored up glycogen down here, and that's going to get broken up and sent through the glucose channel to increase our blood sugar. So again, no blood sugar, pancreas notices that, sends out glucagon to release excess, put sugar back into our blood. So the glucagon binds, GDP is replaced by GTP, which slides down, activates adenylate cyclase, ATP turns into cyclic AMP, which activates a kinase, activates another kinase. This phosphorylation cascade continues to trigger, and enzymes which ultimately phosphorylate the glycogen molecule and break it down into glucose molecules. Those glucose molecules then can diffuse out of the liver cells and back into the blood. So again, transduction pathway has multiple steps involved. But as we look at this, we have reception, we have the transduction, and we have the response. One quick thing I want to point out to finish this thing off is the role of drugs in these cellular pathways. If you see something like this, it blocks part of the pathway. So we have a pathway here, and an increase in AMP inhibits or blocks membrane-bound adenylyl cyclase, causing a reduction in cAMP levels and decreased pKa activity. So again, you don't have to know what all the heck these means, but you have to understand that this is meaning that AMP is increasing and it's blocking this from working. And the drug is metformin. And what is it acting on? Mitochondria. So how does that look in here? Here's metformin, the drug. It We're taking this drug to reduce our blood sugar because we don't want all the glycogen broken down. So metformin goes in, interacts with mitochondria, which increases AMP, which inhibits the adenylyl cyclase. So does it block the reception? No, glucagon will still bind. GDP will still get replaced by GTP. This will slide. However, now it's blocked. Because it's blocked there, we do not form cyclic AMP, and we don't do the rest of the phosphorylation cascade, and the glycogen stays as a large molecule, and the blood sugar does not increase when you take metformin. But doctors are just figured out this new action of metformin. They knew that blood sugar didn't go up, but they didn't really understand where in the phosphorylation cascade that was being changed. So they knew that um, maybe they could make a new drug that would bypass the effect on the mitochondria and then therefore hold off some of those bad side effects. So they're coming up with a new possibility for this new drug. And instead of messing with your mitochondria, maybe we could just get it to do the exact same thing as what the increasing the AMP would do without having to mess with the mitochondria. So this new drug, it would block the adenylyl cyclase because that's where the transduction pathway was blocked. So then we still have that, but then again, we still interrupt the transduction pathway and still reduce our overall blood sugar from stopping our blood sugar from increasing. So again, I know this is a long video, but we're focusing on what happens when we get to the target cell. We're focusing on the reception, the transduction, and the response. Why is the transduction pathway so complex? Because it causes amplification. We can do multiple things from a single signaling molecule. Various responses can occur, not just one response, but it also gives control. There's a smaller chance that transduction might occur in error. So again, hopefully that's helpful. A couple of classic examples: insulin, antidiuretic hormone, and glucagon. And hopefully that breaks down some of that reception, transduction, and response, as well as helps you use those words like phosphorylation and kinase and signaling cascade. Feel a little bit more comfortable with those words. So I hope that was helpful.